everybody. It's another expert talk series. It's another time with your extended family, and we are very grateful to be part of that. As you are aware, research on ALS is continuously progressing. At Everything ALS, we are dedicated to uncovering new discoveries, aiding in early detection, and working towards finding a cure for ALS. You can assist us by participating in our speech study. The purpose of the speech study is to enhance the diagnosis and prognosis of ALS. We are seeking participants to provide 5 speech recordings, each lasting 15 minutes, once a month, over 5 months. It's not that much, but it's super important. We welcome healthy participants as well as individuals diagnosed or probable with ALS to join us. To enroll, please visit everythingals.org/speech project and fill out our form. And you will find a link to that in the chat. And with that, I welcome everyone and will turn this over to Christian.
Thank you so much and welcome. As McFinn said, we are so grateful to have you with us tonight, and we're also grateful to have Dr. Michael Benatar joining us for this expert talk. So I'm going to give a little bit of introduction to Dr. Benatar, and then he'll take over, and we'll have Dr. Benatar provide his presentation and update to the community. And we'll save our Q&A for after that, but please, by all means, during the presentation, feel free to ask any questions you have. We'll take notes of them and provide them to Dr. Benatar at the end of his formal presentation. This portion of the program is recorded so that friends and family can also learn from the presentation. Afterwards, we'll turn off the recording for community-based discussion. So feel free to ask at your degree of comfort, and it'll all be curated after the fact for just the presentation purposes. So with that, welcome Dr. Benatar. Dr. Michael Benatar is Professor of Neurology and Public Health Sciences Chief of the Neuromuscular Division and Executive Director of the ALS Center at the Miller School of Medicine at the University of Miami. Dr. Benatar is the Walter Bradley Chair in ALS Research and the Executive Director of the ALS Center, and he obtained his medical degree at the University of Cape Town in South Africa, and is also trained in both basic neuroscience and clinical research methods. He's been an active leader in clinical and translational research and runs a program focused on biomarker and therapy development for ALS. He is the PI in the ongoing presymptomatic familial ALS study, also known as PreFALS, as well as the CREATE Consortium, a rare diseases clinical research consortium focused on ALS and related neurodegenerative diseases. We are thrilled to have him here, and Dr. Benatar, the floor is yours, and we are ready for you.
Great. Well, thank you so much. I appreciate the warm introduction, and it's nice to see so many faces, some familiar. I'm sure there are others online that I'm not seeing, but thank you for having me and good to see you. I'm going to pull up some slides, and I'm going to give a talk that I hope won't go for too long, although I can be chatty at times. And I'm hoping that we'll have lots of time for discussion. And so what I'd like to do, so I'm just going to rearrange these things on the screen so I can see the slides, and I'll take it down at the end and see you all. So I want to talk to you this evening about something that we've called the Miami Framework for ALS and Related Neurodegenerative Disorders. And I'm hoping that I'm at some level both going to introduce you to some new ideas, and maybe a different way of thinking about ALS and how it's related to other neurodegenerative diseases. But I'm also hoping that this will be sufficiently intuitive that you're going to say to me, this is obvious. But we'll see. And so I'm looking forward to the Q&A and to the discussion. So firstly, these are my disclosures, which I must always do at the beginning of a talk. I do participate in industry trials, most recently sponsored by companies Biogen and Orphazyme. I provide assistance to industry in the design of clinical trials focused on ALS and myasthenia. My research funding is supported from federal sources and patient foundations. And the University of Miami has licensed some of our research data to Biogen to help with the design of clinical trials. So I put this up because this is an open access publication. I'd encourage you, if you haven't downloaded it, to download it and to have a look. I think it's written in a fairly accessible way, and I'm going to walk you through it this evening. And the name of this, the Miami Framework, really just arrives from the location of where we held a workshop, where we had discussions amongst leaders in the community that led to our thinking that forms the basis for this framework. And there's a subtitle here, which is an integrated view of phenotype and biology. And so I'm going to walk you through our thinking about this, what we mean by phenotype, and I want to try to move us from thinking about ALS as a clinical syndrome, something that we recognize phenomenologically or based on phenotype, to thinking about ALS as a biological entity. And I hope that'll become clear through the course of the next 20 minutes or so, what I mean by that. So here's a brief outline of what I'm going to try to accomplish. I first want to talk about phenotypic spectra. So what is the phenotypic spectrum of ALS and what is it related to? I then want to introduce a slightly different idea, but related, of what we might call clinical continua, and I'll explain that. I'm going to talk a little bit about biomarkers, and then along the way, I'm going to show how we build these different elements in to yield the Miami framework. And then I'll try to talk a little bit about what I think the implications of this are for how we should think about the disease and how this might help move us forward in therapy development. So let's start out with one of the ways in which I think our thinking about ALS is changing. And the first is to introduce this idea of a phenotypic spectrum. And this in itself is not a new idea. I think we're familiar with it, but let me walk you through it. So here we're illustrating ALS. And as you know, ALS involves both upper motor neurons, so the motor neurons in the motor cortex that extend down through the brainstem into the spinal cord, and the lower motor neurons that come out from the brainstem and the spinal cord and extend out into the arms and legs and the muscles and control muscle movement. But ALS exists along a spectrum, as you perhaps know, with another disease called frontotemporal dementia, or FTD. And there are different types of FTD. The most common form is known as behavioral variant FTD, or BVFTD, which is illustrated here. But there are other forms known as primary progressive aphasia, or PPA. The important idea behind thinking about these two diseases as existing along a phenotypic spectrum is that patients with ALS may develop FTD. Patients with FTD may develop ALS, and so this is the ALS FTD that exists in between. But in addition, we recognize that patients with ALS may develop cognitive deficits or cognitive impairments or behavioral changes of the variety that we see in frontotemporal dementia or FTD, but not of sufficient severity that we would call this FTD. Likewise, patients with FTD may develop motor neuron dysfunction, but not of a severity or an extent that we would consider it ALS. And so these two diseases really exist along this spectrum. But this spectrum, which we've recognized for a long time, is incomplete. I would argue that there's at least a third dimension to this. And I say at least because I do think there are others. But for this evening, I'll focus on this third dimension, which is what we might call extrapyramidal disorders or movement disorders. Now, the frontotemporal dementia or FTD community recognizes this well because people with FTD may also present with movement disorders that are known as corticobasal syndrome, that's what CBS stands for, or PSP, progressive supranuclear palsy. And patients who present with these movement disorder abnormalities or movement disorder syndromes may also develop FTD. Now, this connection between ALS and these movement disorders is less well recognized, but we do recognize that some patients with ALS can develop CBS or PSP. Some may develop a Parkinsonian syndrome or a Parkinson's disease-like syndrome. Some may develop an ataxia or difficulty with coordination. Some may develop a syndrome like essential tremor, and rarely a Huntington's disease-like phenotype. These are less common, but well recognized and epitomized particularly by patients who carry a C9orf72 repeat expansion. And in fact, the C9orf72 repeat expansion, patients who carry this genetic abnormality are the second most common cause of a Huntington's disease-like phenotype outside of patients who have the traditional or typical genetic mutation that causes Huntington's disease. But these extrapyramidal features or movement disorders are also well recognized in other genetic forms of disease, like VCP, but I would argue even in patients without an identifiable genetic cause of their ALS. So this first part of thinking about these phenotypic spectra provides the first element to how we might begin to build the Miami framework. So we think about this first element as what is the phenotype? And here we think about a motor neuron phenotype, we think about a frontotemporal phenotype, and we think about an extrapyramidal or a movement disorder phenotype. And to help us with some nomenclature, we've called these axis 1 for motor neuron, axis 2 for frontotemporal, and axis 3 for extrapyramidal. And I'm going to come back and build on this further. But let's then talk about a second shift in paradigm in how we think about ALS and these disorders. And this is the idea of a clinical continuum. And so I'm starting out, I'm going to build a figure here, but I want to say that when we as clinical neurologists or ALS neurologists typically think about ALS, we're usually seeing this in what I call a retrospective manner. And what I mean by this is we're seeing patients when they come to us for a diagnostic evaluation. We inquire about symptoms and when they began, and we think of this from symptom onset through the progression of disease as the clinically manifest stage of disease. But because we're looking at the disease retrospectively, we assume that what comes before symptom onset is a pre-symptomatic stage of disease, and we assume that this stage is clinically silent, that there's no outward clinical manifestation of disease. Now, I want to just touch on a few other elements of this figure. We've got diagnosis, we've got symptom onset, and I'm distinguishing symptom onset from disease onset. And I think that's important because I think the biology that underlies the development of this clinical syndrome begins well before symptoms begin. Prior to disease onset, there may be a susceptibility to disease. That susceptibility could be genetic, it could be aging, it could be related to things in the environment, or some combination of all of that. But I'm making a conceptual distinction here between a susceptibility to disease, the onset of disease, the onset of symptoms, and diagnosis. Now, when we study disease and think about disease looking forward as opposed to looking backwards, and we're best able to do this in the genetically at-risk population. So this is something we've been doing now for the better part of 20 years, is identifying and studying people who are at elevated genetic risk for disease, because we want to understand how disease begins and how it evolves during this pre-symptomatic stage. Now, one of the things that we're able to answer is whether the disease is indeed clinically silent during this pre-symptomatic stage. And one of the things that we've learned is, yes, it may be, but patients also go through a stage where there is a prodromal or mild or subtle manifestation of disease. So this prodromal stage represents a stage of disease where there are some clinical manifestations, but they are vague, non-specific, mild, often don't reach clinical attention, but they are discernible when looked for carefully. But they're not of sufficient severity or extent or distribution that we would regard them as clearly indicating ALS. And these may take different forms. When these are in the motor neuron axis, we call this MMI, or mild motor impairment. When they're in the frontotemporal domain, which may affect cognition or behavior, we call this mild cognitive impairment. phenotransition, and I've replaced symptom onset with what we call phenoconversion. So when we're studying the disease this way, we think about disease onset, and initially that's clinically silent. We then think about a transition where the disease becomes clinically apparent, but it's very mild, and it's not clear what the stage represents. And then this disease burden accumulates, and we regard phenoconversion as the transition over into clinically manifest disease. And when this occurs in the motor neuron axis, we think of this as ALS. So let's take this thinking then, and let's extend this a little. So I'm taking you back to this clinical continuum. We had the motor neuron, we had the frontotemporal, and we had the extrapyramidal. And what I'm doing is I'm attaching a stage here of mild motor impairment that precedes ALS. We're attaching a stage of mild cognitive or mild behavioral impairment that precedes frontotemporal dementia. And we're introducing the stage of mild extrapyramidal impairment that precedes these more traditional movement disorders. So if we take this thinking and we now graft this onto the Miami framework, I've already introduced you to these three axes, motor neuron, frontotemporal, and extrapyramidal. And we're now going to introduce this clinical continuum where people may have clinically silent disease, prodromal disease, or clinically manifest disease. And these different stages can arise independently within each of these axes. So you might have clinically manifest ALS, but you might have mild cognitive impairment or mild behavioral impairment that we regard as prodromal on the frontotemporal axis. And you might have a mild tremor that we regard as prodromal on the extrapyramidal axis. And we think it's helpful to think about these in their discrete axes, because these components of disease evolve differently in different people and at different rates in different people, and not every axis is affected in every person. And so we think if we want to fully understand the phenotypic spectrum and manifestation of this disease and related disorders, we have to think across these axes. Okay. So I did want to just say, because some of my colleagues object to the term phenoconversion. They don't like the idea that there is this abrupt transition from a prodromal state to a clinically manifest state. And I will share the skepticism that I don't think this is a hard, discrete point in time. I think that disease evolves along a continuum. The biology that underlies ALS and these related disorders is a biological continuum. But I do think there is value in trying to define what this transition looks like. The best analogy I could give you is to think about age. We think about people as being eligible to vote when they're 18. And we pick their 18th birthday as the point when we say, once you've passed your 18th birthday, you can vote. But I would argue that people who are 17 and 364 days may not be any less ready to vote than those who are 17 years and 365 days, i.e. 18. There's something arbitrary about that cutoff, but we've put that threshold or that transitional state in approximately the right place because we think that that is helpful operationally and for a variety of purposes, like being able to enroll people in clinical trials, for example. So maybe this is more than you want to know, but this reminds me of something known as the Sorites or heap problem. And this is the idea that if you take a grain, say a grain of sand, and you keep adding a grain, eventually you get a heap. But when does that grain become a heap, right? These heaps are of different sizes. This is really a continuous process of adding another grain, and eventually you get a heap, but there's no one point at which you say that grain has become a heap. And I think that ALS, when we think about it as a biological entity, evolves similarly. This is a gradual incremental process whereby there's a degenerative process that's affecting the nervous system. And it's not like a light switch where it turns on or off. You don't have ALS, you do have ALS. This is more like a dimmer switch where the motor neuron system is gradually under greater and greater duress or stress and eventually fails and produces weakness. And that's what we think represents phenoconversion, to clinically manifest disease. So let me then introduce the third idea, which is biomarkers. And biomarkers are really critical to thinking about the underlying biology of disease, right? In the absence of an outward clinical manifestation of disease, the only way in which we know disease is present is by being able to study it through these things that we call biomarkers. So the biomarkers of the underlying biology of the disease are what help us to begin to think about defining disease biologically. So many of you may know that the fundamental pathology in most forms of ALS is this protein called TDP43. Currently, or at least until very recently, we could only detect TDP43 post-mortem by examining the brain under the microscope. Increasingly, we're now developing biofluid biomarkers in the blood that can tell us about the presence of TDP43. Another way in which the TDP43 misfunction or dysfunction is being measured is through this novel type of biomarker known as a cryptic peptide. And these can be measured in CSF and plasma, and we can talk more about this in Q&A if you would like to. Outside of ALS, more relevant to FTD is development of tau seeding assays to tell us about the presence of tau.
The only reason I called was...
Hello? I'll keep going. Another type of pathology in this group of diseases is tau pathology. And in some people who have frontotemporal dementia, for example, it may be caused by tau, it may be caused by TDP43, but in life we typically can't tell. But if we want to direct therapies to the right underlying pathology, we need to be able to make that distinction. So if I come back to this idea, and I've shown you this scheme of the evolution of disease based on phenotype, if we now tack on to this biomarkers, so here I'm showing you neurofilament, which is now well described, and we know that neurofilament begins to rise during this presymptomatic stage, often during this prodromal stage, continues to rise and then plateaus during the clinically manifest stage. This represents a stage of neurodegeneration that precedes the emergence of clinically manifest ALS. But the extent to which we see this change in neurofilament, and the same is true of other biomarkers, may very well depend on the underlying genotype, what the genetics of the disease are, but also very much on the extent to which genotype is just a proxy for the rate with which disease is progressing. When disease is progressing more rapidly, these biomarkers may change earlier. When disease is progressing more slowly, they may arise later. So even for something like neurofilament, which we understand well, we recognize that the temporal relationship between changes in neurofilament and what's going on clinically based on phenotype differs based on the rate with which disease is progressing. And so it's very hard to map these biomarker changes, at least based on what we currently know, into what this clinical progression looks like. And so this is why we built this third element into the Miami framework. I've shown you these axes and these clinical continua, but the other element which we think we need to think about in parallel is the underlying pathology or biology. What are the biomarkers of TDP43 pathology? What are the biomarkers of tau pathology? What are the biomarkers of some of the other pathology? SOD1, for example, in SOD1 carriers who don't have TDP pathology. And then what are the biomarkers of neurodegeneration? And we think that in order to move our fields forward, we need to think, in a sense, agnostically about what the phenotype looks like across these axes and what the pathology looks like, because we don't yet have a neat way to map these onto each other. We eventually would like to. We want to understand what the temporal course is of the pathology through the study of these biomarkers and how that relates to the phenotype. But as of now, our knowledge base is such that we can't neatly map them. And we think trying to do so before we have that knowledge sets us back. And so we're encouraging the idea of thinking about these two in a way that is agnostic to each other. And so another important idea is that these clinical syndromes, MMI, MCI, MBI, on their own are non-specific. If you have mild motor impairment, it does not mean that you will necessarily develop ALS. But as we develop these biomarkers, we think that if people have a clinical syndrome, say MMI, and a biomarker of TDP-43 pathology, for example, that combination would tell us that you are indeed prodromal and that the disease ALS, for example, has begun to emerge. I don't know whether it's a helpful analogy, but I'll draw an analogy from the Alzheimer's world where MCI, mild cognitive impairment, may represent a prodromal stage to Alzheimer's. But typically we think it only does when there's also biomarker evidence of both amyloid and tau pathology. And in the absence of those biomarker evidence of pathology, that MCI may simply reflect an aging phenomenon or something unrelated to Alzheimer's disease. So in order to really understand what the meaning is of these prodromal states, we have to expand our understanding of the pathology, which we do through the development of these biomarkers. So let me then try to summarize what I think the Miami framework hopes to encapsulate. It proposes a classification system that comprises two parallel components. The first is phenotype. And within phenotype, there are at least three parallel subaxes: the motor neuron, the frontotemporal, and the extrapyramidal. And then within each axis, we recognize a phenotypic continuum, from a clinically silent stage of disease to a prodromal stage to a clinically manifest stage of disease. The second parallel component is these biomarkers of underlying pathology and the biology of neurodegeneration. And we think as of now, we need to think about these in a way that is agnostic to the underlying clinical phenotype. And we think that this framework offers a unified approach to specifying the pleiotropic or very diverse manifestations of disease, and also will help us develop an understanding that we can describe the pattern and trajectory of these emergent biomarkers as we learn about them more. So let me give you a few examples just to illustrate how this can be practically applied. And I'm going to illustrate this with patient A. And think about this as a patient who carries an SOD1A4V mutation. This, if you don't know, is actually a very aggressive form of disease. Once this becomes clinically apparent, patients with this form of disease may only live 6 to 12 months. So what we know about this form of disease is that people may first develop mild motor impairment, and then they may phenoconvert to clinically manifest ALS. They may, depending on the SOD1 mutation, then develop mild cognitive impairment, but they typically don't show any manifestations on the extrapyramidal axis. And in somebody with this form of disease, which is very aggressive and evolving very rapidly, we would expect the neurofilament light level to rise during this clinically silent stage of disease, to continue to rise during the prodrome, and then to plateau during the clinically manifest stage. By contrast, for example, patient B may have a C9orf72 repeat expansion mutation. This person may first develop mild behavioral impairment, then mild motor impairment, and then might phenoconvert to clinically manifest behavioral variant FTD or BVFTD. They may well then develop a tremor as a manifestation of the extrapyramidal axis, and then may evolve ALS and have FTD-ALS along with a tremor. And if this is evolving slowly, as it often may, the neurofilament rise may only occur at this stage of disease. And so what I'm trying to illustrate is that the phenotype in the phenotypic spectrum varies based on genotype, or even amongst people who don't have an underlying genetic cause of disease, and the temporal course of the biomarker will differ based on that different biology, related either to genetics or to the rate with which disease is progressing. So what then are some of the implications of thinking about this framework? So the one is, I think we need to change the way we think about our diagnostic criteria. You may know that the Gold Coast criteria are currently how we think about the diagnosis of ALS, and these require progressive weakness or impaired motor function alongside, and both signs of upper motor neuron and lower motor neuron dysfunction. But when we're following people, for example, who carry a pathogenic variant, we know that they may develop weakness with either upper motor neuron trouble or lower motor neuron trouble. And the challenge, if you apply the Gold Coast criteria, is you'd have to wait until both of these develop to call them ALS. And we think that that will simply lead to a delay in diagnosis and a delay in therapeutic intervention. And so in the context of somebody having an identifiable genetic cause of disease, where the likelihood that weakness and signs of upper or lower motor neuron dysfunction represent ALS, we would favor calling ALS and making that diagnosis sooner so that we can get people into clinical trials. And so we would say that we should hearken back to some of the principles in a previous version of the diagnostic criteria, the LS-Scorial criteria, to help promote that early diagnosis. Here's another implication. At the moment, the frontotemporal dementia community who think about MCI before FTD and the ALS community who are thinking about ALS with mild cognitive impairment define this cognitive impairment very differently. And that's problematic because we lead to different definitions and disagreement about what represents disease and what doesn't, and how do we communicate that to people who may or may not be affected. And so we think we need a unified approach to understanding and describing these prodromal stages of disease, because we think it's critical not just to our understanding, but to what we're communicating to people who are participating in research studies or coming to a clinic who may have very early manifestations of disease. And I think that one opportunity here is for the ALS and FTD communities to work together to harmonize their definitions of these prodromal states. So what then are some of the implications of this framework? So the one is, I think we need to talk, and I'll be interested to hear your thoughts about how we communicate the presence of these prodromal syndromes. Do people want to know that we think they have a prodromal state? And if they do, how do we communicate it? Well, there are challenges. As I've told you, different groups have different definitions of these. And so there's not one definition. And so if we're communicating to an individual person that they have one of these syndromes, there's a degree of uncertainty around that because the definitions of these are not fully agreed upon. The other thing which I've highlighted is that these prodromal states are not always prodromal to manifest disease. Without the biomarkers, they may not represent an early stage of ALS or FTD. So the interpretation is unclear. So if we're communicating to people that they have MMI or MCI, we have to be able to communicate the uncertainty that or the uncertain significance that that carries, that may or may not portend an imminent future of ALS or FTD developing. And there are socio-legal considerations. When these diagnoses, and I put that in scare quotes, are communicated in a research context, we can keep that out of the medical record. But if that gets into the medical record, that may impact things like long-term disability insurance. And so we have to think about those protections when we begin to talk about communicating this information to people and how this is documented in the medical record. So I think the approach we need to adopt is informed consent, to partner with our community, to communicate what we know, what we don't know, what our uncertainty is, and to understand individual preference for whether they want to know about the emergence of these states and what they may or may not represent, and then make sure we have the right infrastructure in place to help support people in the journey of navigating the uncertainty of what that might mean. I think we also have to now develop biomarkers that reflect the underlying biology of disease, because these will be critical to overcoming that uncertainty and giving us greater confidence as to what these prodromal states might mean, both for people's future risk, but also potentially to enroll people early in clinical trials to try to prevent the emergence of clinically manifest disease and to overcome this long-standing problem we have of this long diagnostic delay in ALS. So let me try to wrap up, and then I'm pleased we're going to have a good amount of time for some discussion. So I think the Miami framework offers a new conceptual understanding of ALS and its relationship to other neurodegenerative disorders. What we're trying to do here is encourage a shift to thinking about disease based on a biological definition, rather than thinking about this disease only as a clinical syndrome, recognizable as what has been described in textbooks for years as ALS or FTD. part of the move or the push towards thinking about ALS and defining it biologically is we're then going to increasingly focus development of treatments on that understanding of the biology and recognizing that the biology may be different in different people with disease, and that a therapy needs to target the biology that's relevant to an individual who has that biology, rather than an individual who might have the clinical syndrome, but a different underlying biology. So the Miami framework also recognizes this underlying biological continuum and the value of recognizing these different clinical states. We think this can facilitate earlier diagnosis and the potential for earlier therapeutic intervention. And I think it encourages transparent communication with our unaffected carrier and patient community about what we know, what we think we know, what this might mean, and really sort of partnering in figuring this out together. So before I stop, I'm just going to acknowledge our participants who participated in our studies, their families, my colleague Joanne Wu, with whom I run our research program at the University of Miami, as well as our research team, and then all of the colleagues and representatives from the community who attended the workshop in Miami about 18 months ago that led to the genesis of these ideas, as well as all of our funding agencies. So I'm going to stop there and look forward to some dialogue.
Thank you so much, Dr. Benatar. That was super informative. And it really goes to show the need to understand ALS also in a greater context within this family of neurodegenerative afflictions, and certainly this desire that we all have to understand at a much deeper level the kind of the root causes of what's going on. We do have a few questions from the audience. So I'll jump up and ask the first one on behalf. Really as a follow-up, could you please comment on whether there's a potential role of digital biomarkers and or digital outcomes in defining the biological stages? We certainly are using digital cognitive assessments and other tools. So wondering what you could offer in that front.
Yeah, so I think digital tools tell us something about the phenotype, right? It's a way of telling us, for example, in your speech study, are there elements in speech or the use of language that tell us about an outward manifestation of disease? That sort of by definition is part of the phenotype. But I don't think a digital marker is going to tell us what the underlying biology of that is. Is that TDP43? Is that SOD1? Is that FUS? Is that tau? I don't think that the digital marker will speak to the biology, but it may be a useful tool for phenotyping.
Thank you. Let's see, I've got another. Go ahead. Go ahead, Indy.
Yes. So thank you very much for the great talk, Dr. Benatar. And the next question was, one of the person who actually is in the audience had their brain scan for their mother. And one of the things was what they found was the frontal part of the brain was dead. I'm not sure how you can actually find from the scan. And she had she lost her ability to speak and lost her control for her movements. So would that consider kind of the FTD or the cognition? Is this one of the ways that you actually detect through imaging?
Yeah, so I think there may be several parts to your question, Indu. So the first is, I think that imaging is one approach it's one biomarker approach to understanding this disease. So, imaging may tell us about the structure of the brain. It may tell us about the function of the brain. There are different imaging modalities that can shed light. I would say as a general rule in both ALS and FTD, our imaging methods have not yet lived up to their full potential. And a big part of that, I think, is that they are relatively noisy, and our signals come out of group level analyses. And so we often have to study groups of people and say this group of people are different to, for example, a healthy group. That has value, but it's less immediately relevant to being able to tell an individual you have the emergence of disease, for example, because there's so much overlap between what a brain scan looks like in somebody with ALS and somebody who doesn't. I mean, traditionally, routine clinical imaging, the brain scan doesn't look any different in an ALS patient to somebody who doesn't have ALS. And so the brain MRI is not traditionally a useful diagnostic test other than for ruling out other things. That's traditional, usual imaging. There are more refined techniques that can give us greater insights, but again, they tend to be more helpful at a group level. Just one other thing related to your question. Cognition, again, is a phenotype. It's something that we measure. I would never say somebody has a cognitive problem or deficit based on an MRI scan. That's a clinical judgment. It's like I wouldn't say you're weak based on what your EMG looks like. You're weak because I test your strength and your muscles are weak. So they tell us different things. But you are right that structural abnormalities, especially in the frontotemporal lobes, may be the anatomical correlate of what we see in patients who have frontotemporal dementia. So I hope that sort of answers the question.
Thank you.
Very much. Thank you. I'll take the next question. And this one came in via private channel. How would researchers determine whether the Miami framework is more or less effective than other ways of thinking about ALS stages? And how would you decide if these ideas are helpful?
It's a good question. So I don't think right now there are other proposed ideas of how to think about the disease prior to the clinical manifestations of disease. The traditional thinking is that this is a clinical syndrome. Many of you may have been through the experience when symptoms first developed that you saw a neurologist and they didn't know what to make of it. And they might have done some tests and they might have said, come back and we'll see what happens or how this progresses over time. Or while you were going from doctor to doctor, the disease progressed until it became very apparent. And that's the old way of thinking about this. And we can keep thinking about the disease in this way. But the truth is that has left us in a state where, despite everything we have done, the average duration from symptom onset to diagnosis is 12 months. That's in the US, that's in Europe, that's worldwide. That's notwithstanding all efforts we've made to develop biomarkers. I think it's fundamentally a problem in how we think about disease. We sort of say there needs to be so much the disease before we're willing to call this ALS. It needs to be so widespread or so manifest. And what we're pushing for with this idea is to say, if we begin to think about what the earlier manifestations of the disease might look like, yes, that's going to put us at a point in the disease when it's less certain. But if we then study people at that stage and develop those biomarker tools of biology, that's going to help us identify disease at an earlier stage. So we have no guarantee that this is the only approach or the right approach. But I think it's a useful approach that puts our foot in the door, that gives us a framework for thinking about this and for designing studies in a way that can help us understand the disease more. And I have no doubt that as we learn more about the disease, this will evolve and this will need to be modified. I mean, I already said to you, I don't think they're just 3 axes. We've called those out because those are the dominant axes that we see. But there are people, for example, with some genetic forms of disease where the muscles may be involved. So we may say we need a muscle axis. And so I think that one size may not fit all, but what we're trying to do here, I think, is push us towards thinking about disease biologically. And I will just say the Alzheimer's field is well ahead of us here. The Parkinson's disease, the Parkinson's with dementia, the Lewy body disease is increasingly thinking about disease in this way. They want to do away with terms like Parkinson's and Parkinson's with dementia and Lewy body disease. They want to talk about neuronal alpha-synuclein disease because alpha-synuclein is the pathology that underlies all of those diseases. Now, we can't talk about TDP-43-opathies. We could, but not all ALS. Most of it, but not all of it, is a TDP-43 pathology. The FTD community certainly can't because half of their disease is TDP-43 and half of it is tau. So I think we have a more varied biology than just alpha-synuclein or just mutant Huntington in Huntington's disease. But if you ask me, I think the future is that we will all be talking about this disease biologically. We're just not there yet. What I'm trying to do with this framework is nudge us along that way to sort of thinking a little bit ahead of the curve in how we need to think about this disease. And I'll say one more thing, which I said already. And all of you know this, that I don't think that ALS is one biology. I think it's many different biologies, and that biology may be different at different stages of disease. And we are still stuck in the old way of doing clinical trials, where we put everybody with ALS into one clinical trial with a drug that works in one way. And it's probably the case that that can at best only help a subset of people. But to the extent that we begin and increasingly think about ALS biologically, we're going to be driven to saying, you have biology A, you should get drug X. You have biology B, you should get drug Y. And I think that's the future of where we want to go with therapy development. And until we think about the disease biologically, we're not going to get there. So I've been strident, but I also want to again come back to the humility of the question that I don't know this is the right framework, but I am confident that we have to think biologically, and that as we do, that will shape our thinking about what the phenotype looks like and what we call it and how we describe it. And there is sort of a virtuous cycle between the clinical phenotype, the biology, the clinical phenotype, the biology. And as we learn about the one, we learn about the other. So think about how we started with ALS, and then we started finding genetics. And then we said, gosh, ALS and FTD may be related, so we found C9. Once we find C9, we recognize there may be non-ALS and non-FTD manifestations of C9, so we update the thinking about phenotype. So this is how we refine our understanding of disease in medicine and generally. We cycle between the clinical observations and then probing the underlying biology. And that helps us to refine our clinical thinking, which in turn helps us to get closer to a biological understanding. So I think this is by its nature an iterative process, and this will need to be refined in the future.
Thank you. Thank you for the
Sorry, long answer to a short question.
No, this is great. I mean, you're a great storyteller, so that's very easy to absorb in. So thank you. The next question that we had was more on, can you tell about the involvement of APOE4 gene, as you talked about Alzheimer's, in diagnosed FTD and ALS diagnosis? Have you seen those, coexist, or they're completely not?
So I'm going to have to beg ignorance on this. I do not think of ApoE as an ALS gene or a risk factor for ALS. I do not think of it as an FTD gene. But what I will say is that neurodegenerative diseases don't exist in isolation. There's often co-pathology. And when you look at the brains, for example, under the microscope, and Dr. Ostro's here, he can tell us more about this, but you often see mixed pathology. You see Alzheimer's pathology, and you see TDP pathology, and you see some vascular pathology because blood pressure and cholesterol and other things catch up with us as we age. And so that's another element actually to the Miami framework that we can think about, is it can accommodate the idea of mixed pathology that is going to vary in proportion in different people. And Lyle, I hate to call upon you, but I wonder if you want to add something since you're more knowledgeable about this than me.
So I don't know that I'm more knowledgeable about it than you, Michael, but I think your answer was perfect. I think what we could say qualitatively is it definitely isn't the same sort of thing as in Alzheimer's, right? In Alzheimer's, ApoE4 really matters, right? And we can tell you, that's just not the case. Now, I think maybe what you were sort of suggesting, getting at is, and this is not my area, but in Alzheimer's, there's a suggestion that certain ApoE alleles cause certain biological problems, like with cholesterol or lipids. And I know there have been some studies in ALS and in FTD that suggest that, if you have those same ApoE alleles and you have ALS or you have FTD, yeah, you might also have a problem with cholesterol or lipid handling. And so, this idea of overlapping biology seems to be true, but there certainly is not the same kind of effect on, disease risk or how old you are when you get the disease as there is in Alzheimer's. Is that helpful?
Thank you.
Yeah.
Very.
Right, thank you. So I'll jump in with another one. Could you please clarify how you define lower motor neuron weakness in this framework when proposing diagnostic criteria? Does lower motor neuron weakness require a diagnostic EMG, or can the EMG be non-diagnostic or normal in combination with what we're talking about here?
Great question. I forgot I wasn't talking to a group of neurologists, so I have to explain myself. So I think about the motor neurons or the pathology in ALS as involving two types of neurons. There's the upper motor neuron and the lower. And the way to think about that is like a two-person relay race, right? You're familiar with the 4x100. 4 people have to run to get around the track. For a signal to get from the brain to the muscle, 2 runners have to run, the upper motor neuron and the lower motor neuron. So when the upper motor neuron is sick, what we typically see is a slowness of movement, a stiffness of muscles, what we call a spasticity. We see reflexes when we test the reflexes that are very brisk. These are the traditional manifestations of upper motor neuron. How might that manifest subjectively? Typically as a slowness of movement, maybe a clumsiness of movement. If that is affecting speech, the speech becomes slow and drawn out and strained because the muscles are not moving quickly and swiftly. The lower motor neuron, by contrast, leads to a loss of muscle tone. The muscles become floppy, like wet, limp noodles. The muscles waste away. They become atrophic. The reflexes disappear. Weakness is tricky. Weakness can come from lower motor neuron trouble and upper motor neuron trouble. I would venture that most of the weakness in ALS comes from lower motor neuron trouble, but with a contribution from upper motor neuron. And neurologists tease those apart by looking at the distribution of which muscles are weak and what's the relative strength and weakness. So that's a hard thing for one subjectively to know where weakness is coming from. But I would say overwhelmingly, weakness itself is mostly coming from the lower motor neuron. I hope that answers the question without getting into too much excruciating detail.
Thank you.
That's a very useful explanation. Thank you.
The next question is, if somebody is a carrier of C9, but asymptomatic, but does have, much muscle twitch once in a while, what are some of the early indication of onset that they should be thinking about? And also, are there any kind of trials that's going on for C9 that they need to be aware of?
So great question. So firstly, just to say that muscle twitching, fasciculation, is a normal phenomenon. We have probably all had it. Now, I understand that when you carry a pathogenic variant that puts you at elevated risk for ALS, every fasciculation has a different meaning, and you think and you worry about it more. But by and large, isolated fasciculations we do not think of as an early manifestation of disease. The same is probably true for cramps. We worry about fasciculations and cramps when they come with wasting of the muscles and when they come with weakness of the muscles. And so I would say that by and large, one should not worry about isolated fasciculations here or there. These are normal phenomena, again, that can be exacerbated by sleep deprivation, dehydration, caffeine, some medications. There are lots of things, exercise, that can bring out fasciculations. As of now, the only pre-symptomatic trial that is going on is really the SOD1 trial of tofersen in SOD1 carriers. We are not yet at the stage of having pre-symptomatic or prevention studies for C9 carriers. My hope is that those will come, but we're not there as of now.
Thank you.
Thank you. So here's also a little bit more of a general question, but it speaks to so much coverage that you've provided here, which is how can so many different biologies cause this one very specific disease of ALS? And how can all these different biologies cause different diseases?
It's a great question. So I'm going to pull some analogies again. You might have heard of something called anemia, a low blood count. Who's heard of anemia? Yeah?
Yeah.
Okay, good. So I'm not talking Greek. Okay. So anemia has many different causes. You can be anemic because you're iron deficient. You can be anemic because you're vitamin B12 deficient. But the symptoms of anemia to you, fatigue, lethargy, shortness of breath, pallor, they all look identical. But the biologies are fundamentally different, and you cannot treat B12 deficiency with iron supplementation, and you cannot treat iron deficiency with B12 supplementation. So we know that this happens in medicine and in biology. It happens all the time. And the anemia is a great example of how we have to move from the phenomenology or the phenotype to understanding the biology if we want to bring the right treatment to the right patient. Now, we don't yet understand in ALS how a different biology leads to the same phenotype, but I'll venture an idea that there might be multiple inputs that ultimately lead to the same problem. So think about a clogged highway. You get a traffic jam. You could have a traffic jam because there was an accident. You could have a traffic jam because there's road work. You could have a traffic jam because, I don't know, there was a protest on the streets getting onto the highway. All of those produce a traffic jam, right? But the cause is different. And so we have to think about ALS in the same way. In this analogy, the traffic jam is the motor neurons, the upper motor neurons, the lower motor neurons degenerating. But what causes that? There may be many different causes or inputs that can lead to that. Now, we know this already. We know that SOD1 mutations are not the same as C9, are not the same as NEC1 or TBK1. These are all different genetic mutations that lead to a phenotype of ALS that we can't differentiate. I can't look at you and tell you your ALS is C9 and your ALS is NEC1. I can't tell that difference clinically. So we already have examples in front of us where very different biologies produce the same phenotype. So I don't know how it happens, but I know for sure that it does. What I also know is that we're not recognizing it enough in people in whom there isn't a genetic cause of disease. So I know I've only given you a partial answer, but I don't have a better one.
No,
Thank you.
those were really good examples. Thank you.
So the other question is, what method of testing is needed to detect TDP-43 solely in the cytoplasm?
So currently we do not have a test. So we're just beginning to get biomarkers of TDP pathology. They're in their infancy, but they don't tell us where TDP-43 is. Is it in the cytoplasm? Is it somewhere else? But I'm going to give you a little caveat to that. I mentioned this thing of cryptic peptides, and I don't want to get too far into this, but let's just talk a little bit about this. One of the things that happens with TDP-43 in ALS is TDP-43 should normally be in the nucleus. And in the nucleus, it has a role to play in regulating how genes are spliced together, what's spliced out and what's spliced in. When TDP-43 in ALS or in FTD is in the wrong place, i.e. in the cytoplasm, not in the nucleus, there's what we call a loss of nuclear function. So TDP-43 is no longer playing the role that it should be playing in the nucleus in regulating that splicing. And one of the things that can lead to is a missplicing that leads to what's called a cryptic exon and maybe a cryptic peptide, which is a new sort of protein or mini protein that is formed because of that sort of inaccuracy or lack of fidelity in that inclusion-exclusion during that splicing process. And then we can detect that as a biomarker in the biofluid. So if you had to ask me, as those tests become better, something like a cryptic peptide might be a marker not of necessarily of TDP-43 pathology, but of loss of nuclear function of TDP-43. So maybe we'll develop other assays. When I say we as a community, this is not me, this is other people doing this work, we'll develop biomarkers that will tell us about different aspects of TDP-43 pathology. You know, maybe the loss of nuclear function is what comes earliest. Maybe the cytoplasmic aggregates come later. Maybe, you know, there's a different sequence and different biomarkers of TDP-43 pathology will tell us different things. But again, we're at the relatively early stages of having those tools, and those biomarker assays need work for us to really be able to rely on them.
Thank you.
Let's see, we've got quite a number of questions left. I will offer two things since we're running a little bit short on time. Those questions that are study specific, we do have we are lining up community updates from those companies and their experts to be able to provide those updates. So we may not get to them tonight as we address some of the other questions from the audience, if that's okay. You agree with that?
Yes.
Okay, thank you. So here's a question as well. Should ALS patients be tested for dementia periodically? I'm a caregiver and my friend was scared a year ago, and at that time we had the brain scan done. There was no evidence of dementia or Alzheimer's. How can this work into the care paradigm with ALS as well?
So it's a great question. First, I would say a brain scan doesn't tell us about the presence of dementia or not. I alluded to that earlier about the role of MRI. I do think that all patients with ALS should periodically have some clinical screening for cognition and behavior to know whether there are early manifestations of a frontotemporal syndrome. The frequency with which that should occur is not clear. I will say that the American Academy of Neurology guidelines for the care of patients with ALS, so these are quality guidelines, do emphasize the importance of this. I would also say that I think ALS clinics in general don't do a good enough job, us included. And part of the challenge is that it takes time to do that adequate screening. And ALS clinic visits are already often long. They take several hours. And so we and others are trying to come up with ways to help develop better screening tools. But I would caution us against doing perfunctory screening where we're doing something because it's quick, but not because it's actually helpful or sensitive in telling us whether there's a problem. So I think there's work that needs to be done in this space, but I do think it is important.
Thank you so much.
Thank you. And we have heard so many people say they love the presentation and the way you explain. One of the big questions is, how can we help you?
How can you help me? I think you can spread the word. I think we need to get these ideas more out into the community. I'd encourage you to ask questions about this and push other speakers. When you see your neurologist, push them. I would encourage you to participate in biomarker studies. I know that we all want to participate in clinical trials and they're critically important. But our clinical trials, we have a long history of them not succeeding, not developing, not delivering on their promise. And I believe a big part of that is that we don't understand the biology of disease. So participate in clinical trials, but also participate in observational studies that are focused on biomarkers, because I truly believe they're critical to us breaking through in terms of, one, developing therapies, but also developing earlier diagnosis and even paradigms of ALS prevention. So that, I guess, would be my shout out. Get involved in those studies and help.
Thank you. Thank you for your time today, and also excellent presentation. And we will actually put this on YouTube this weekend, and I'm sure you'll get a lot more people watching this we have audiences from 60 countries. So thank you.
Excellent. Thank you for having me. Lovely to see you all, and have a good night.
Yeah, thank you.
Michael.
Bye.
Bye-bye.
What a pleasure. There you have it, folks. Another presentation coming right to your living room from one of the most dedicated research doctors around. This is why we think that you should spend your time with us, because we really spend a lot of time to bring these programs to you. And we feel like we're doing our job, but we also feel like you're part of the solution. There's a lot of work that the doctors and researchers do, but there's also opportunities for us, especially like we have our speech project. It's something very easy to do and means so much. Was that something?
I'm going to jump in real quick, McFinn, because we did have a question actually about the speech study, which is, does it help to participate in the speech research if one is taking Nuedexta? Everybody helps, right?
Yes.
Absolutely, everybody is a help.
Absolutely, yeah.
Boom. I was hopping up and down when I saw an earlier question too about what's the point of participating if there's nothing can really be done right now? And, I want to invite obviously Indu and your experience and McFinn and the Pathfinders, because I think it comes from you best. But I know personally, my many years now of working with all kinds of rare diseases and so passionate about being with you here, that this is how we get diagnosis earlier. This is how we get more shots on goal, is participating in the studies that drive understanding of everybody's experience. But I don't want to speak for everybody else who has such a deeper experience with it. So I call on the community to answer that one.
Yeah.
And let's start with you, Indu. How do you feel about that?
Yeah, I mean, I think this is why we do what we do, right? I mean, the whole citizen-driven research is very, very important, because we, the people, have to drive. I mean, why we're doing the citizen-driven approach is because otherwise it's too slow, and it just, the data, the research trickles down as opposed to us coming together and let's collect the data and share with everybody else. So I would say please get enrolled in our speed study and also the asymptomatic study. So if anybody's a gene carrier but do not have the ALS diagnosis, that's a great way to participate. And we are actually coming up with more studies so that we can, give the data back to you as well.
That's a great.
Yeah, folks, for you to understand where you're at is a big, I think it's mentally really healthy. So it's by becoming educated about what you feel most worried about. And that's one of the reasons why you come to this platform here is, what are you worried about this week? Because I'm sure that there's somebody else worried about the same thing. And that's why we come together, so we can lower the stress level.